Hello, in this video we'll be talking about blue-white screening. As the name suggests, it's a screening process. After 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 cloning the gene of interest into certain bacteria, we use this particular blue-white screening strategy to select our recombinant colonies. So if we walk through the workflow of cloning, we would understand the necessity of doing a screening. So first, in order to do the cloning, you need to restriction digest your vectors with restriction enzymes. Then you have to also restriction digest your gene of interest with the uh, same restriction enzyme. The next step would be the ligation of these two things in order to get the recombinant plasmid. Once we get the recombinant plasmid, then we can we can transform that into a competent bacteria. So bacteria would eat up the plasmid, and as we grow the bacteria would grow in number, and so as our gene of interest would grow in number as well. But there is a problem. If we think about the culture system, there could be one possibility that the transcription efficiency is the transformation efficiency is low, and that is why some of the bacteria has not taken out the plasmid. But this problem could be solved easily. So some of the bacteria have the plasmid, some of the bacteria might not have the plasmid, but the bacteria which has the plasmid has the antibiotic resistance gene as well, right? Now, when you are preparing the media, you're putting antibiotic into the media. So if the bacteria don't have a antibiotic resistance gene, it would die. It would simply die. It cannot survive in that environment. Only if the bacteria has antibiotic resistance gene, it can survive. Without the antibiotic resistance genes, it cannot survive. So this problem is solved. But there is another problem. The second problem is now in the solution there could be two type of plasmids. Like one is just self-ligated plasmid, non-recombinant, and something there could be like a recombinant plasmid. Now, how to understand that the bacteria colony that is formed have the recombinant one, not the self-ligated one? So in order to do this, you need some screenable markers, and very often the screenable marker uh, is present in the form of a particular reporter gene. In this case, the reporter gene is LAGZ. Now, LAGZ is present in the multiple cloning site. Now, if we clone our gene of interest in the multiple cloning site, the LAGZ reporter gene would be kind of perturbed because we are inserting into the locus of LAGZ gene, so its product won't be formed. So LAGZ normally give rise to beta galactosidase. Now beta galactosidase would convert XGAL into a colored product. So that would make the bacterial colony looking blue. Now, if you insert the gene of interest into that particular LAGZ containing multiple cloning site, then what would happen? Beta galactosidase won't be produced because LAGZ gene is already disrupted. So this is called insertional inactivation. Due to the insertion of your gene of interest, the LAGZ gene is inactivated, and as a result, beta galactosidase is not produced. So the XGAL cannot be cleaved and it cannot produce any color. As a result, the bacterial colonies which have this kind of recombinant plasmid, it would have white. But at the end of the day, there could be two different type of plasmid. So in a plate, you would have some colonies which are blue and some colonies which are white. So remember, the white colonies are those colonies which are recombinant, because in these colonies the bacteria has the recombinant gene, and due to the insertion of the gene, there is an inactivation of the LAGZ. That is why these colonies are white, and that explains why do you need a screenable system and why it is useful. And once you can see and understand that okay, the white colonies are recombinant, you would pick up those colonies and culture them in bulk. Once you cultured them, you can isolate the plasmid and keep the plasmid in -20 or -80 degrees centigrade for future use. So this was the overall view why a screenable marker is important, or what is actually blue-white screening. So this is widely used screening method in molecular biology. I hope you enjoyed this video. If you like this video, give it a quick thumbs up. Don't forget to like and share and subscribe.